AQA finally released an official abbreviation list in February 2026, containing abbreviations you are allowed to use without writing out in full. And this has left me wondering, will there be stricter mark schemes in summer 2026 examinations? This supersedes previous guidance where we were told if the abbreviation appears in the spec, it can be used as an official abbreviation. Now, there are some surprising omissions from this list, so keep watching to ensure you don't lose marks from a possible increase in mark scheme strictness. In this video, we will review the official list from AQA and refine our understanding of the terms and when we are allowed to use them. Then we will test your recall and spelling of many of the important terms. Why do I need to spell them if I can use the abbreviation, I hear you say? Well, official guidance from AQA say that in the essay, the full word is needed in the first instance. Then the abbreviation can be used in the remainder of the essay. Does spelling really matter? Well, for some molecules in particular, a misspelling introduces significant scientific inaccuracy. For example, students often write ATP as adenine triphosphate instead of adenosine triphosphate. Missing the ose suggests a lack of understanding that there is a ribose sugar in ATP, as well as the adenine base and the 3 phosphate groups. That one error can cost you 5 marks in an essay by capping your mark. 2 of these errors could potentially cap your essay at 15 marks, regardless of how amazing the rest of your essay is. It really depends on the error and the use of the term as to how important the spelling is. Now, the official AQA list was published in alphabetical order. However, we are going to work through them in groups of relatedness. This will allow us to ensure we know the differences between tRNA and siRNA, and PCR and VNTR, and be able to define the terms also. Our first group is our core biochemical and molecular abbreviations. We will run through the official abbreviations allowed first, then review some not on the list we should be aware of. ADH can be used for antidiuretic hormone, ADP for adenosine diphosphate, ATP for adenosine triphosphate, and CAMP for cyclic AMP. Make sure you know what each of these molecules do. Now we will move on to our nucleic acids. DNA can be used for deoxyribonucleic acid, cDNA for complementary DNA, which remember is DNA made from a messenger RNA template using reverse transcriptase. RNA can be used for ribonucleic acid, messenger RNA, which carries our code from DNA to ribosomes. Our ribosomal RNA, we can use rRNA. tRNA is our transfer RNA that has an important role in translation. Then we have siRNA, our small interfering RNA, and RNAi, RNA interference. These short RNA molecules silence gene expression by inhibiting translation. Pause the video if you need to make a note of the differences between these nucleic acids. Now we'll move on to those not on the official list and must be written out in full. We'll start with rDNA, our recombinant DNA. Remember, this is DNA made from combining DNA fragments from 2 different species, and the word species is really important when giving this definition. mtDNA for mitochondrial DNA is not allowed. Remember, mitochondrial DNA is circular DNA found in mitochondria, inherited maternally, with no histones or introns. Then TF for transcription factor we cannot use. We must write out transcription factor in full. Remember, these are proteins that regulate gene expression by controlling the rate of DNA transcription into messenger RNA. They bind to promoters to activate or repress recruitment of RNA polymerase. Next, we will review some abbreviations related to biotechnological processes. Here are authorised official abbreviations we can use without writing out in full. ELISA for the enzyme-linked immunosorbent assay, PCR for the polymerase chain reaction, GM for genetically modified organisms, IPS for induced pluripotent stem cells, VNTRs for variable number of tandem repeats. Now we will review some key abbreviations from the AQA list related to metabolism and photosynthesis, starting with those allowed and moving on to some very important omissions. So first of all, we have GP for glycerate 3-phosphate, our 3-carbon compound formed in the Calvin cycle. RUBP for ribulose biphosphate, GPP for gross primary productivity, and NPP for net primary productivity. Make sure you can define all of these terms and make sure you know what the different molecules do and where they appear in cycles. Moving on to our coenzymes. Remember, a coenzyme is an organic, so carbon-containing, non-protein molecule that binds to an enzyme and helps it catalyse a reaction. They carry chemical groups or electrons between reactions and are temporarily changed but regenerated later. They work alongside enzymes to process metabolic reactions. So the abbreviations we are allowed are for NAD and NADP. NAD is a coenzyme that carries hydrogen and cycles between oxidized and reduced state. In glycolysis, NAD is reduced when triose phosphate is oxidized to pyruvate. In the link reaction, NAD is reduced by pyruvate. And in the Krebs cycle, NAD is reduced whilst oxidizing the 6-carbon citric acid to a 4-carbon molecule, oxaloacetate. This produces 3 reduced NAD per cycle. Our coenzyme NADP is reduced in the light-dependent reaction when receiving hydrogen ions after chemiosmosis. It is then used to reduce GP to triose phosphate. Now let's look at those that are not on the approved abbreviations list, and I think these 3 are going to catch students out the most. TP is not on the list as an approved abbreviation for triose phosphate, so you must always refer to it as triose phosphate in your answers. The abbreviation hasn't been accepted in mark schemes for a few years now, and it still catches some students out. Remember, triose phosphate is produced from the reduction of GP in photosynthesis and used to build glucose or regenerate RUBP. In the newly released official abbreviations list, there is no NADH abbreviation either. So whilst in the past it has been commonly accepted in mark schemes, to be safe, I would always write reduced NAD and not use the abbreviation NADH. Similarly, the coenzyme NADP, which is reduced in the light-dependent reaction when receiving the hydrogen ions from chemiosmosis, does not have the abbreviation NADPH shown in the official list from AQA. I would therefore avoid using the abbreviation NADPH and refer to reduced NADP in case this is a deliberate omission and a change in trend in stricter mark scheme specificity. Interestingly, FAD has not made it into AQA's official abbreviation list either. Whether this is an oversight, I'm not sure, but best to write it out in full if referring to it. Here are some other terms that haven't made it to the official abbreviations list that we've got to make sure we write out in full. LDR for light-dependent reaction, ETC for electron transport chain, acetyl-CoA for acetyl coenzyme A, ES for enzyme substrate complex, and Vmax for maximum rate of an enzyme-controlled reaction. Now we will look at some important chemical groups. These abbreviations have made it to the list. PI for inorganic phosphate group, COOH for the carboxyl functional group in organic molecules, notably amino acids in our specification. RCOOH, the general formula for a carboxylic fatty acid. Just remember the R refers to the hydrocarbon chain that's of varying length and saturation attached to the fatty acid. NH2 is our amine group and can be used. The next group of abbreviations we're going to look at are linked to physiology and coordination. So those that are on the official list and the abbreviations that can be used without writing out in full are SAN for sinoatrial node, AVN for atrioventricular node, and IAA for indoleicetic acid. Now, beware using these abbreviations as they are not on the list, but we might commonly try to use them. CNS especially for central nervous system, PNS for peripheral nervous system, and HR for heart rate. We must not use these abbreviations. We must write out those terms in full. Now we will look at molecules involved in immunology. We are allowed TC, abbreviation for cytotoxic T cells. Remember, these kill host cells showing foreign antigens, and these could be infected cells or cancer cells. We're also allowed TH for T helper cells. Remember the 4 roles that T helper cells have: activating B lymphocytes, producing cytotoxic T cells and T memory cells, and stimulating phagocytosis. And then we have HIV, our human immunodeficiency virus. Remember, this virus attacks the T helper cells. Now, here's some immunology terms that interestingly didn't make the list. AIDS for acquired immune deficiency syndrome, so spell it out full to be sure. Remember, in HIV infection, viral load spikes initially but then drops in what is termed clinical latency, where there's no symptoms seen as T helper cell levels remain high enough. It's when the T helper cells drop below specified levels that AIDS symptoms appear. We also don't have APC for antigen presenting cell on our approved abbreviation list, nor have we got AB for antibody or AG for antigen. Our final group of terms on the recently released official AQA list are some special symbols. So alpha and beta, we are allowed, and you might be using these to describe the different isomers of glucose or your alpha helix or beta pleated sheet in proteins. You can use the symbol for chi-squared when naming the statistical test. Remember, this test plays a significant part in CPAC-10 investigating responses of organisms. Then we have our water potential symbol. Water potential is the measure of water's tendency to move from one area to another. Pure water has a water potential of 0, and water will move towards areas of more negative water potential. You will lose marks if you refer to concentration of water. SD for standard deviation is not on the list and must be written out in full. And lastly, the symbols for ions, for example Na plus or Ca2 plus, are accepted, but the charge must be correct. So now we've seen which abbreviations we're allowed, let's look at when we're allowed to use them. You're allowed to use them without writing the term out in full when answering general questions on papers 1, 2 and 3 that are not the synoptic essay. However, if your answer contains an accepted abbreviation from the list, but then you also name it in full incorrectly, then the abbreviation will not be accepted. AQA are also very clear in their document that in the essay, the full word is needed in the first instance, then the abbreviation can be used in the remainder of the essay. And lastly, if the command word is name, make sure you write whatever you are asked to write out in full and do not use the abbreviation. Now it's time to test your recall on some of the important abbreviations we have discussed. First of all, can you spot the abbreviations on here that would not be allowed in general question answers without writing out in full first? Pause the video and decide for each if they would be allowed. Now check your answers. The abbreviations in green are official abbreviations which can be used directly except in the essay. Those in red must be referred to in full in all questions. Now let's check your recall of the spelling of some of these important terms to make sure you get the marks in the essay or any questions where the command word is name. Again, pause the video, write out the terms, and then we'll check your answers. So let's check your spelling carefully. So pause the video again and mark your answers. So that's our review, refine and recall of the official AQA abbreviations document that was released recently. We'll just have to wait and see how this changes our mark schemes this summer. But at least we are now prepared. You know what abbreviations you can use and when you can use them. If you want to prepare yourself further, check out Biotechnology for related content.